So I'm Deb, also from Newcastle, like Steve, so we have similar introductory slides. And I work on mainly the childhood cancer neuroblastoma, but I'm also the director of the VIVO Biobank, which you've heard about has provided lots of biospecimens for the research advances that we've been discussing. So for anybody that isn't aware, neuroblastoma, and I know you'll be parents from lots of different childhood cancer types, but neuroblastoma sounds like it's going to be a brain tumour, but it isn't, because it's a tumour of the sympathetic nervous system, which is this sort of nervous system. Oh, the pointy thing doesn't work. So it's this nervous system that runs either side of the spinal cord between the neck and the groin. So not within the central nervous system, the brain and the spinal cord, it's either side. And as a result, you can get a variety of symptoms that can occur from sometimes pressure on the spinal cord, but equally the commonest site for it to occur is the adrenal medulla. So young children will present with abdominal masses, or sometimes you can get problems with constipation or problems with neck masses. And if it's spread, then it can be more like leukemia in terms of getting sort of anemia and bone pain and things. And one of the reasons why I've always been interested in it was when I came to Newcastle long ago than I care to remember, from having worked in the US. My mentor at the time was very interested in neuroblastoma because of its heterogeneity, and I call that a chameleon effect. And it soon became apparent that because of the ways in which it can present in any of those locations, but also importantly because of its biology, you could really see how this tumour was changing its spots either in response to chemotherapy or sometimes by itself, and could sometimes escape detection. And what I'm showing here on this slide is what it looks like under the microscope. So you can see that it's one of the small round blue cell tumours of childhood. And what you've got here is these blue cells because of the dye that's used to stain them, but producing this sort of fine angel hair or neuropil, which is characteristic of a neuroblastoma that's poorly differentiated. In other words, it's a nerve cell that's trying to become a normal nerve cell, but it can't quite really because it's too immature. What I am pleased to tell you is that over the last 40, 50 years that CCLG has been in existence, since the 70s, we have made advances in childhood cancer and in neuroblastoma, such that most children now do survive, and that's something in the order of 65%. However, it's not true for all children. And this is one of the reasons why I've always been interested in neuroblastoma, because despite the advances that we've seen in genetics and treatments, which I'll come on to, unfortunately, the largest group of children with neuroblastoma is this high-risk group, which is about half of children. Say in the UK, that would be 50 children a year, as opposed to these other two groups, which are 50 children who do quite well, thank you very much, by and large, apart from one small group I'm going to talk about later. And we stratify these groups on the basis of age, because neuroblastoma is very much a tumour of young childhood with a median age of presentation of around 2, and very rare in teenagers and young adults. And so we know that age is a determining factor of prognosis, together with whether the tumour is spread or not. And then also, importantly, this genetic marker, which is an oncogene, a cancer-causing gene called MYC, which was first discovered about 40 years ago now, and still plays as major role as it did back in the 80s, and as it does now. And I'll tell you why that is a bit later. So in order to diagnose neuroblastoma accurately, not only do you need to look at it under the microscope, but you've got to do the fancy genetics, some of which Joan and Steve have talked to you about, and which include this sort of imprint slide, something called fluorescent in situ hybridization. And I'm going to tell you a little bit about genetics because I think it's so important. Your normal cell would have 2 copies of every chromosome. So one from mum, one from dad. And what you find in a cell that's got the MYC gene that's amplified, it's present in hundreds of copies. You know, this is big time, way too much of a dosage of a gene for a single cell. And what that does to a neuroblastoma cell is it lets it divide and really get out of control. So patients that have MECAN amplified neuroblastoma, which is around 20% of all neuroblastoma, but about half of high-risk neuroblastoma, will have rapidly proliferating tumours, something a bit more like lymphomas or leukemia because of this MECAN gene. And that's why it's still so important then as it is now, because it will tell us not only how fast is the tumour dividing initially, but how well it might respond to treatment, because if you divide fast and you're growing fast, then you shrink fast as well. But that's not all. That was 40 years ago for MECAN. And so we've made lots of other advances with genetics, including looking at other genes. And I'm going to tell you a little bit about one called ALK. I'll tell you a bit more about that later, but it is possibly getting up there in terms of importance for neuroblastoma as MECAN. And these are the different ways that we can test ALK within a lab. And as Steve said, for medulloblastoma, we have a similar national genetics reference laboratory in Newcastle for neuroblastoma, which was in existence for some time now. So we worked through a battery of genetic tests, starting with the MECAN fish, looking at the chromosomes in general and what's called a SNP array, looking for ALK, looking for lots of other genes that might be targetable, that have got mistakes in them. And then we also can send a tumour directly for the testing, which you may have heard of, called whole genome sequencing, which is even more detailed look at the tumour. And then finally, we bank it within the VIVO Biobank. So I'm going to home in a bit on high-risk neuroblastoma because that's the one that we've still got to address most of our efforts. And as I'll just quickly recap, that's neuroblastoma that could be MECAN amplified, as in here, where there's two cells with lots and lots of copies of MECAN, or it's neuroblastoma that's spread in a child over the age of 12 or 18 months. So that's distant spread to either a bone marrow, this is a clump of neuroblastoma cells in bone marrow, or to bones, and this is an MIBG scan, a specialised scan that takes up neuroblastoma around the body, and they're the two most common sites of metastatic disease. I'm just highlighting here some additional genetics, really to show you these genes that I'm very interested in, like MECAN, ALK, and another cancer-causing gene called MGM2. And this is a copy number plot where you see these spikes here, it tells you that the genes are present in too many copies, so not doing, causing cancer in ways at which they shouldn't do if there was only two copies. But I also want to highlight another way in which we've been able to stratify neuroblastoma, which is on the basis of whether you have changes in the numbers or parts of chromosomes. So we all have 22 what we call autosomes, so normal chromosomes, and then an X and a Y chromosome. So we have the 23 in total. And what you find is that if you've got sort of poor biology neuroblastoma, as in high-risk neuroblastoma, you tend to have these sort of alterations in segments where half of the chromosome might be lost or half might be gained. And it's something called a segmental chromosomal abnormality. And it's become another really important test in addition to MECAN, such now that, particularly in infants and babies, we use the presence or absence of these segments of chromosomes to determine how we treat them, because we know that for some babies you can get away with not giving any treatment if there isn't MECAN amplification and you've just got losses or gains of whole chromosomes. But then continuing on the theme of neuroblastoma, what's really sort of revolutionised cancer biology and diagnostics in the last 5 years for particularly adult cancers and now childhood cancers, including neuroblastoma, is this concept of these circulating tumour cells. And if ever a tumour lends itself to detection of circulating tumour cells, it has got to be neuroblastoma. And the reason being because it's more likely to invade bone marrow and bones than many other childhood and adult cancers. And so what happens is the main tumour, which is this bulk of cells here, lots of different colours reflecting lots of different mutations, this genetically heterogeneous or chameleon type appearance. These tumour cells will break off and go into the bloodstream where they'll be in amongst the white blood cells, and then they'll depart from the bloodstream into, say, a bony site, or in the case of bone marrow, we call them disseminated tumour cells. And over the last 5 years, we've worked out ways in which we can detect these in the blood of children with neuroblastoma and use them to find out important information about their genetics, how the tumours might respond to treatment. So what we did with some funding that we had from the Little Princess Trust a few years ago for a PhD student was to take a number of patients with neuroblastoma of all risk groups, and we analysed their blood samples and bone marrow samples on this fancy bit of kit called an imaging flow cytometer. So not only does it measure the numbers of these circulating tumour cells, but you can also visualise them. So you might think, well, how do we visualise them when they look, just like cells, and how can you tell that they're not white blood cells? Well, you can stain them for particular markers of what are white blood cells or neuroblastoma. So in this case, for these circulating tumour cells, we stain them for GD2. And some of you may have heard of GD2 because it's also an immunotherapy given to patients with neuroblastoma. And importantly, we exclude white blood cells by staining those for CD45. And so we can measure the numbers both in the blood and the bone marrow. And these graphs here tell you that the sort of numbers that we see in children with neuroblastoma of circulating tumour cells in particular far exceed numbers that you would see in many adult cancers. So almost, neuroblastoma is sort of a solid tumour leukemia in these cases. And then we also did some work to show that in patients with high-risk neuroblastoma, the numbers of circulating tumour cells correlates, this is at diagnosis, correlates with how well these patients might clear their bone marrow at the end of induction chemotherapy, because normally we don't know. We give patients induction chemotherapy to get rid of metastatic disease, but we can't really tell until we do the scans and the bone marrows at the end of induction to know if we might have cleared them. But this suggests this measuring the numbers of circulating tumour cells might be useful to help predict how well these children respond to high-risk neuroblastoma treatment. So then the new kid on the block, sort of, is not just circulating tumour cells, it's this concept of circulating tumour DNA, which in some ways is probably more manageable than looking at the circulating tumour cells, because, as I say, you need more fancy bits of kit for the circulating tumour cells. For the circulating tumour DNA, you can get a regular blood sample. It goes into a regular tube that you would use for measuring other sort of blood chemistry. You spin it and you take off the tumour DNA, which is part of circulating free DNA. And in some ways, there's nothing novel about these circulating free DNAs. They've been used to test for fetal DNA to sort of diagnose any fetal abnormalities for a long time within the obstetrics departments. But within cancer, they came in particularly again in adult cancer and now in paediatric over the last few years. And what we find is these very short segments of DNA, we can get all the genetic information, or at least most of it, that we can get from doing an invasive tumour biopsy. So these have so much more advantage. Oh heck, I'm running behind here. So much more advantage for monitoring of disease, picking up new biomarkers, and importantly, can we detect disease using this circulating tumour DNA before it starts to become apparent in patients developing symptoms or by scans. And as I've said here, you really pick up the chameleon nature because the circulating tumour DNA can have many different mutations at different times. And because I'm coming to the end, I'm not going to be able to tell you about the other part of the research, but I'll just carry on about the high risk for now. So we looked at the presence of ALK circulating tumour DNA. So ALK is another oncogene. It's a bit different from MECN in that MECN does its job in the nucleus, whereas ALK is sort of bound to the cell membrane. And when it's attached to its sort of lock and key, it will stimulate the cell to undergo a proliferation, but from the outset. And it can be either mutant, as in mistakes, or it can be amplified, a bit like MECN, in around 10% of neuroblastomas at diagnosis, but importantly, over a quarter at relapse. And why is that so important? Well, as you might have guessed, there's now new targeted therapies against ALK, not developed for neuroblastoma, but developed for non-small cell lung cancer. And they have made their way into the clinic for patients with neuroblastoma, so it becomes such an important thing for us to look for. We've shown that having an ALK aberration is associated with a poor prognosis, and so the plan is now to introduce ALK inhibitors into patients at diagnosis, whereas currently we're doing it at relapse. And I'm just going to show you some work that we did using some plasma samples. This is a particular case that we found an ALK mutation. Well, we actually had a master's student that found 2 ALK mutations. Well, why is that important? Well, they can be slightly different and they might respond to slightly different ALK inhibitors. And we looked at diagnosis in this patient and found one was present at a high fraction, about 30%, and one was present at a low fraction, about 3%. And then when the patient had their tumour biopsied, only one of the other mutations was present. So it looked like there was a difference between the mutation in the primary tumour versus that in the plasma. Now, sadly, when the patient went on to relapse, there was another sample taken, not only where the patient relapsed, and that showed only one of the types of ALK mutation, but also when we did the plasma at relapse, we found it was the same as the metastatic site. And what that tells you is that the particular ALK mutation that went on to cause the patient to relapse was the one that was actually mainly circulating in the blood at diagnosis. And that tells you that actually getting plasma or circulating tumour cells is more effective at the most malignant clones, and therefore perhaps targeting treatments towards what's present in the blood or the bone marrow is going to be more important for the patient. And this is just showing you that in this particular patient, there was also plasma taken at the end of treatment, which showed no evidence of mutation, but sadly 3 months later, when the patient relapsed, that ALK mutation was present. Okay, so I'll do this last slide now on the journey of patients with high-risk neuroblastoma. This came from one of our Click Sergeant social workers at Newcastle, and I think it depicts very well the tortuous track that patients take, the very intensive treatment, and the fact that it can have lots of ups and downs. So beginning with chemotherapy, if you're lucky, you go into remission, but if you don't, you have what's called refractory, and then you need other treatments. But hopefully you can then respond and have surgery, and then the patient's going to have the high-dose chemotherapy, which is fraught with its own problems, which can be anything ranging from lung or liver problems, but hopefully they get over those and then have radiotherapy, and followed by the new treatment that came in. Well, it's not new now because it's been around for a long time, immunotherapy. But it's at this stage here, as you're going through this treatment, that sadly we know that approximately half of patients with high-risk neuroblastoma will need to come off their current planned treatment and begin relapse strategies. And that's why we really need to sort of be working out which patients are going to relapse, how can we pick them up earlier, and what can we do to help that. And even when patients have completed treatment, as Sue Carey, our social worker, said, it's very much leaves its legacy of the worry of relapse. And so one of the things we've done, again with Little Princess Trust funding, and this is pretty much my last slide now, is to look at the epidemiology, so characteristics of patients who relapse and whether we can learn about particular features, what are the clinical and biological features. And this goes back to the MYC amplified patients, where if you take MYC amplified relapsed neuroblastoma patients, we've learned from our study and others that sadly MYC amplified ones have a much more aggressive course at relapse. And that's important because if you're working out which treatments to give, you want to make sure you give the right treatments and you're not comparing necessarily the effect of the treatments in patients who have MYC amplification at those that haven't. So this is just ongoing work that we hope to publish very soon. And then finally, we've also found out, and again, it helps our understanding of relapse, and we need to think about how we incorporate this into trials. But for patients with high-risk neuroblastoma that do relapse, we know that those that relapse within the first 18 months of diagnosis, so that's sometimes they may still be on treatment or they might have finished it depending on how many delays they've got. They do worse than those patients who relapse after 18 months of treatment. And sometimes, as a result, you can have quite a lengthy period of stability, but that's less likely to happen if you're an early relapse. So I think I better call it a day there. I was going to tell you about low and intermediate risk, but I haven't really got time and I don't want to stop you all from having lunch. So I'll just quickly skip to the end where I want to thank everybody who's been involved in this work, all of our funders, VIVO Biobank, and very happy to answer any questions about neuroblastoma or VIVO Biobank over lunch. Thank you.